Hi everyone. In this video, we will be discussing the ideas that revolve around the concept, the central dogma of biology. So what does it mean for it to be the central dogma of molecular biology? This means that DNA can be replicated or like copied, and it can be transcribed into RNA and basically um, be made into protein from RNA. It can also go back from RNA to DNA through a process called reverse transcription. So we'll be talking about each of these methods, including replication, transcription, and translation. And so the first thing is DNA replication. It occurs in the S phase of the cell cycle, like we were talking about, with mitosis and meiosis. DNA replication is semi-conservative in that what it means is that there's a parent strand and a new strand that comes together every time DNA replicates. And whenever this occurs, synthesis occurs from the 5' end of DNA to the 3' end. So in this lecture, there will be some videos like this. Make sure to actually watch them. They will actually help you throughout like the homeworks and stuff like that. So major enzymes that are involved in replication, it includes the DNA helicase. This is basically a little enzyme that basically goes in and unwinds the DNA and opening it up into a replication fork, and I will show you what that means in a few seconds. And it uses ATP to basically make that cut throughout, allowing the strands to kind of swivel freely around the other strand itself. Then there are what is called single-stranded DNA binding proteins. These proteins basically bind to the DNA strands that are separated and keep them from rejoining back with the hydrogen bonds that exist. So these come in between the two strands, or the hydrogen bonds will end up coming back together. Then, so one thing that the replication process can't do is that it cannot start off out of nowhere. It needs a short sequence in which it allows you to have a starting point. So that is called a primer, and the enzyme that creates this primer is a DNA-dependent RNA primase, which basically makes that little RNA primer to kind of bind to the first few nucleotides within that template DNA. Now, there's the DNA polymerase. This is the enzyme that actually adds the nucleotides to the end itself. We'll talk about the steps in the next slide. That way it makes more sense. But this is what this does. It goes through and it adds on nucleotides as it goes on. Then so this is particularly for DNA polymerase 3. And it goes from there Then there's DNA polymerase 1. This basically removes the RNA primers and fills the gaps by base pairing, inserting new nucleotides to replace that primer itself. So polymerase 3 extends the chain. Polymerase 1 removes the primer and inserts the rest of the parts of the DNA for that. So it's like a repair enzyme, but it's required in this scenario. Then there's DNA ligase. DNA ligase is basically a sealing enzyme in that it joins any gaps that were created because of this whole thing. There's especially when it's like not covalently joined, basically it's ligating everything together, so it's like gluing or sealing it. In bacteria, it usually ends up with the phosphate bond of the NAD plus as an energy source. Some of them actually use ATP, depends on the system itself. So when I was talking about replication forks, this is what that means. So it's like this fork here, where it's like two strands that become opened up by helicase, and the single-stranded binding proteins keep them apart, creating a fork in which the polymerases can go through and set up their way. So the steps include helicase and the single-stranded binding proteins, opening and stabilizing the DNA strands, priming the sequence with RNA primer to start off DNA synthesis through RNA primase. And then it starts adding on the different DNA nucleotides through DNA polymerase 3. And then it removes the primers using DNA polymerase 1. And then through exonuclease activity, that just means that it just takes out the primers through an activity where it just takes out the primer itself, basically chewing it up and degrading it, basically. Then there's the joining of fragments of DNA, where it's like these, where, so one thing about DNA replication is that it can only go from the 5' end to the 3' end. So whenever it goes in the opposite strand, where it's technically from 3' to 5', what happens is that it goes in fragments where it's like 5', starting from the 5' end, making different fragments, because they're not actually like continuous, because they have to create a primer every time they start, as they kind of go through the chain this way from 3' to 5'. And what happens is that it generates a set called Okazaki fragments. And these Okazaki fragments end up being joined together by DNA ligase. DNA ligase is basically gluing these separate components together. Now, in most cases, the energy itself comes from deoxyribonucleotide triphosphate monomers, or DNTP monomers. The polymerase 3 is what actually is involved in chain elongation, and also has editing abilities that allow 3' to 5' exonuclease activity, which means that it will go through and add on what they need. Typically, it would probably make a mistake, 1 in every 10 to 9 base pair, 10 to the 9th base pair. So that's a big, big number. Very less chance. But when they do make mistakes, they do end up fixing it in like proofreading abilities of these polymerases themselves. So now we're gonna go into transcription. Transcription itself is basically making an RNA copy of the gene to basically use the template itself, using the DNA as a template itself. So what this means is that this is now the time where we're basically going into how different genes that we have are expressed, and it's basically transcribing the language of DNA into RNA. That way the cell machinery can read it and be like, okay, this is what we need to do this kind of thing. So basically transcription occurs in the nucleus for eukaryotes and the cytoplasm of the prokaryotes. So again, this is another video. It will help you guys understand transcription in a more thorough way. This is more of an overview in lecture to make it a bit easier on you guys. So make sure to watch these videos as you kind of go through the slides themselves. So transcription itself is the synthesis of an RNA. It is the first step in expressing a gene itself. So when we have this, it requires different DNTPs, including a uracil triphosphate, uridine triphosphate. So all of these are the different DNTPs, a DNA template, and an RNA polymerase. Within a gene, there are different regions. We'll talk more about this in a little bit as well, where there's a promoter, there's a transcription initiation site, like this one here. And then there's the RNA coding sequence, and then a termination sequence. So that's basically what a gene itself is looking like for the most part. And what happens is in transcription, these DNTPs are required, a DNA template, and an RNA polymerase instead of a DNA one. And in this video, we're kind of going to talk about the differences between these two, between prokaryotic and eukaryotic transcription, where it can vary depending on how the gene is structured, how kind of goes in. So before we kind of consider what the genes themselves look like, or how they're expressed, we need to understand the different differences between the two different types of genes for prokaryotes and eukaryotes. So in prokaryotes, the structure basically starts off with like a promoter region. This is a specific DNA sequence that is upstream, or like before the start site of the gene. Then there's the start gene, start site, where it's for transcription. And it goes through this entire RNA coding region. There is no separate parts to this. And there's a termination sequence that comes up and it terminates it. And then everything after that is like very downstream. The promoter itself is a signal that gets the expression of the gene itself promoted. So it basically is like signaling it to be like, yes, please express this gene. Now, similar things with eukaryotes, there is a promoter, there are the coding sequence here, and a termination region. But with eukaryotes, it's a bit more complex in that eukaryotes are primarily more complex than prokaryotes, in that they have some enhancer elements. These are more regulatory elements here, enhancer and proximal ones. So these are basically helping it control how it's expressed. So if it's like enhancing it or inhibiting it, that's where those components would be. Then there's that promoter region, just in case if it's like we need this occurring. And then we have what is called exons and introns. So these exons are primarily non-coding. Exons are the coding regions of the DNA. The introns themselves are non-coding for the most part. Some people like to call it junk DNA, but at the same time, it's primarily just that we don't know what is actually happening with these introns. And then there's a poly A sequence to kind of just like keep it more stable. So in prokaryotes, what happens is that the core polymerase itself that comes in for transcription is made up of 5 different subunits, 2 alphas like this, beta and beta prime, and omega that comes within this as well. And this polymerase binds to what is called a sigma factor, this one here. And what it does is that basically makes a holoenzyme. And that holoenzyme is basically going through the sequence. And it's like, okay, let me find this specific region that I want and start off. Once that specific sequence is found for this polymerase, the holoenzyme, what happens is sigma gets released, and this core enzyme here, this core polymerase that has all these different subunits, ends up shooting in the direction of transcription, where it's like going it transcribes it, basically adding RNA nucleotides to your RNA sequence itself. It goes on transcription, and then what happens is it hits a area where it's basically having a kind of a stop kind of signal. It's kind of row dependent in this case, and like this sequence here. And once it hits the sequence, it ends up stopping the entire process of transcription in prokaryotes. Sorry, this is row independent, this is row dependent. So it kind of comes in, kind of stops it in its tracks like this. Now with eukaryotes, there is the same 3 steps: initiation, elongation, and termination. It starts off with this Tata box here, where it brings in a couple of transcription factors or transcriptional activators where the initiation complex gets formed. So the transcriptional activation factors, those come in, and like the Tata binding box, the Tata box binding protein comes in and it binds to form that initial complex. It involves 3 different RNA polymerases, but in transcription, we're going to focus on RNA polymerase 2, which is primarily for mRNA or messenger RNA. And if it's like, so each of the different types of RNA polymerase has its own function in which type of RNA they produce. RNA polymerase 1 is ribosomal RNA. The polymerase 2 for RNA is mRNAs. And then RNA polymerase 3 is where tRNAs get produced. So now, once all of these different factors come in together, they're like, okay, all the correct polymerase is in. We have some other transcriptional factors like 2B and 2F. Like in this case, it started off as 2D, and then it kind of went in. It comes together, and what happens is it starts elongating the process. And what happens, it comes in and adds more of them, and the initiation complex itself is added on. And what happens is the polymerase itself goes through the sequence until it reaches a region like this and cleaves. This region is like a termination site, similar to what we see in like prokaryotes. And what we see is it cleaves like 11 to 30 nucleotides away from it and starts polyadenylating it, because, or what that means is just adding on many A's like this, except no U's, just A's throughout. So now, what do we have right now? So now we know that we can replicate DNA, so we can have many copies of them. You can even do this in a test tube through a process called polymerase chain reaction, which is basically a technique in labs that you can make like millions and millions of copies of the same DNA region. You guys may have done this when had like COVID tests and stuff like that. So that's another thing. That's how it occurs. Basically, you're replicating the viral DNA on that viral RNA. And then now we know that we can also transcribe it into mRNA, but this particular type that is directly out of transcription is called pre-mRNA or the nascent transcripts. So how do these become more mature? This goes through an idea called RNA processing. This is basically trying to remove any introns, right? So with prokaryotes, we already know that there are no introns. So RNA processing does not get involved in this scenario. But then when, because the thing is with transcription and translation, it just occurs all simultaneously. But with eukaryotes, on the other hand, it's a bit different, just because when RNA has to be exported from the nucleus, because transcription and RNA processing occurs in the nucleus, because you want the RNA or the pre-mRNA to be a bit more stable, and removing out like the introns can be a bit more helpful in creating the desired protein properly. And the thing is, to do translation itself, the mRNA has to be exported into the cytoplasm for translation to occur, and it must be modified. So what is the process that is allowing these RNA to be modified before translation? And that is called alternative splicing. Alternative splicing is basically in ways that it can be spliced. So splicing is basically removing any introns in between. And what happened, it can also depend on how it's being spliced. So if these colorful ones are your exons and the in-between spaces are your introns, these arrows is kind of like showing where it's being spliced. And what we see is different combinations of proteins depending on how it's being removed and put together. So it ends up with many, many different types of proteins from the same gene by using this process called alternative splicing. So now this is with now it brings us to translation, where it basically makes our proteins from the gene that basically, like, this is how it ends up being expressed in your system. It's like it basically makes the protein according to like the transcribed instructions on the mRNA. And what we see is that with eukaryotes, it's on the ribosomes. In the cytoplasm, for both cases, it's in the ribosome, in the cytoplasm. Make sure to watch this video. It will actually help you all. So what is translation and what does it require? Translation itself is the process of converting different information in the molecule itself into a sequence of amino acids that makes up a variety of proteins. It requires ribosomes, transfer RNAs, amino acids, and energy. So like forming peptide bonds and just going through that process. So ribosomes themselves, they are made of both protein and ribosomal RNAs. Basically, when translation occurs, the large and small subunits of the ribosome come together. It holds the mRNA in place. And what happens is that the tRNAs just go through each of the different sites, starting from the A site to the P site to the E site, and we'll talk about those and it kind of just keeps on adding on to the chain itself. So what does this ribosome do? It basically helps create a scaffold in which tRNAs and mRNAs can be brought together to basically use enzymatic activity to form peptide bonds. So remember when we were talking about proteins in the first week, where peptide bonds are formed between the monomers of amino acids to form these primary sequences and so on. And what happens is with prokaryotes, it's the 70S ribosome where it's 30S and 50S. It comes together And what happens is that this is the structure itself. It has a couple of uh, ribosomal RNAs that kind of just like allow some activity to occur. And then there's the 80S ribosome where it's the 40S and the 60S components together with a couple of other ribosomal RNAs. And basically what happens is it kind of becomes the way of decoding and using tRNAs, where it has a specific amino acid that is covalently attached to its 3' end, right? The amino acid's up here and binds to that mRNA at the codon to decode the triplet code. So this code here, it's basically trying to decipher it, and basically it folds into a cloverleaf secondary structure like this. And the anticodon loop, this loop here, is what determines what codon it deciphers. So if you have an example of this, where it's like AC 5' CCA, it pairs with a 5' UGG. And what happens is it codes for tryptophan, which we'll kind of cover in this here, where it involves the genetic code itself. So one thing about this is how researchers back in the day were like that the 4 letters of DNA or RNA language can be used to basically determine 64 different 3-letter word combinations called codons. So it's like reading another language here. So what happens is there are 3-letter words basically with these, like in mRNA language basically. And what they did was when the protein encoding gene is read, the codons themselves only like code for like 20 amino acids. It's basically added in the protein sequence and in what order. There is one start codon that is called methionine. It's the start of the protein encoding sequence in the mRNA itself. Then there's 3 stops, UAA, UAG, and UGA. Those are ochre, amber, and opal. Those 3 are stop codons. This is basically starting off the process of termination in the mRNA itself. And then the remaining 60 codons just basically either code for a particular amino acid, and you can see that's there can be multiple codons that code for one um amino acid itself, like leucine here. And reading this, you start off with this side here, the first base, if it's like what is, so for example, if we have like U, you would go U, 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 right? So like we have first base, second base, and then we find the third base. And in this case, it would be phenylalanine. Phenylalanine, sorry. So that is the genetic code. It basically goes from mRNAs to proteins in amino acid codons, basically. So now a couple of things that you guys need to know about the genetic code itself is that it is nearly universal. All codons specify for the same amino acids in all organisms with a few minor changes among organisms. It's redundant in that all amino acids except two of them are encoded by more than one codon. But it's unambiguous. One codon never codes for any for more than one amino acid. So it can have multiple codons coding one amino acid, but it's never that these can code for multiple of them. So it is also conservative in that when multiple codons specify for the same amino acid, in most cases, the beginning two letters are usually identical, like this example here, where valine has GU for the most part for the beginning part, and like proline where it's like CCs, threonine with ACs, so on. So now, how can we actually use this code? When we use this code, what happens is you can know the DNA sequence. You can know if you know the DNA sequence, you can predict the codons that come with it. If you know the amino acid sequence, you can approximately predict the mRNA or DNA sequence that comes with it, depending on what you have. And this kind of allows you to kind of sort through any mutations that appear, because sometimes there could be issues in like a different diseases where you end up with the defective protein, and that protein ends up like, okay, let's correct this. How can we correct different scenarios like this? And in some cases, it could be like the techniques could be like molecular cloning or CRISPR gene editing. It kind of comes into that idea of like trying to resolve this scenario. So that kind of helps you kind of predict what is actually happening. So now with translation, it also has the same three steps: initiation, elongation, and termination. In prokaryotes, what happens is there is a initiation complex where it involves a small ribosomal subunit, couple of initiation factors, and mRNA at the codon, AUG. And then once that kind of comes together, there is a region called the Shine-Dalgarno sequence that it kind of comes together. It binds with the 16S ribosomal RNA, and what happens is that it basically scans the area for this region. And basically base pairs with that ribosomal RNA in the 30S subunit. Now with eukaryotes, what happens is there's this 5' cap, which is the 7-methylguanosine cap, like this. And what it does is it goes the 40S ribosomal subunit just goes through this sequence here from the cap, and it's like da 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 da. Ah, yes, we found the initiator. Their region, which is the AUG codon. And then slowly each of the different other components come together and gets into like a initiation complex, which kind of starts off bringing the initiator transfer RNA, and it kind of goes through that process to set up for, it brings in the initiator tRNA, and then rest of the subunits come in, like the exit sites and the amino acyl sites, and it kind of just brings in the larger ribosomal subunit. So now what happens in elongation for translation? So the tRNAs, they kind of bring the amino acids one by one to kind of add to this chain here. And what they do is they start off by coming in. It adds on this chain by using energy. It kind of just like binds there through peptide bonds, because it's the A. So there's the amino acyl site, which is the A site. It binds the charged amino acyl tRNAs, which basically contain amino acid. The peptide area is where it basically makes it binds and forms peptide bonds. So peptidyl site is for P sites or for peptide bonds. And then there's the E site where it basically releases the uncharged or dissociated tRNAs, that they can be free to basically charge with, get recharged with free mRNA, free amino acids. So now it kind of goes through this process until it hits like a stop codon. Once a stop codon is hit, it basically starts like with these nonsense codons, where it's like these 3 codons, and when there is no complementary one, a release factor or this protein here kind of comes in and binds to that stop codon. It basically stalls the ribosome, and what happens is it ends up stopping the process, and everything gets dissociated and frees the tRNA and the polypeptide. And that polypeptide basically is the primary structure of the amino acids, like the proteins. So that's that amino acid sequence is the primary sequence, and then it goes into like the alpha helix and the beta sheets, which are the secondary structures. And then there's the quaternary, which is the 3-dimensional shape, and then multiples of these kind of come into the quaternary structure of proteins. And with that, I will conclude this course. I hope you folks all had fun. And yeah, this talk is now the end of it.